So I'm going to start with the introduction. So welcome everybody to the seminar for today. It is my absolute pleasure to introduce Professor Martin Giurfa. He's a French-Argentinian neuroscientist. His position is the exceptional class professor of neuroscience at the University of Paul Sabatier in Toulouse. He obtained a PhD in neuroscience in the University of Buenos Aires in 1990, and then he moved to Germany for a postdoc. And he worked with Dr. Randolph Menzel, who was a real pioneer in this use of bees as an animal model for trying to understand how memories are encoded in a brain. Bees are known for being very smart, and so it was a very useful model for this task. He spent 11 years in Germany, where he actually became an associate professor of the Free University of Berlin. And then in 2001, he moved to Toulouse in France and became a full professor of neuroscience. And there he created the Research Center for Animal Cognition at the French Research Council, and the institute that he directed for from his creation until 2017. Then, well, currently he's a senior member of the Institut Universitaire of France. So this is the Academy of Science in France. He is also a member of the German Academy of Science and the Royal Belgian Academy of Science. He has received multiple awards. One of them is the Silver Medal of the CNRS. He has like over 200 papers in many high impact journals, and his research focuses on perceptual phenomena and learning and memory in insects. But really, his contribution to the field is really important because before Martin research, people thought about insects as biological machines, really, like robots that they respond to stimulus, and so they have this stimulus response dynamics where they have very little or any cognitive abilities, and the people thought that you know, these high order cognitive abilities were restricted really to mammals or primates and humans, and then insects were just like machines. And his research has really changed completely the way we think about insects, and he has provided compelling evidence that insects are capable of high order cognitive tasks like learning of concepts. They can count, they can resolve very complex tasks, and he really has, you know, he's been the spearhead on this, you know, change in the paradigm of how we think about insects. So he's really the authority in this field, and it's my pleasure to, you know, have you here, Martin. Thank you, welcome and thank you for accepting this invitation.
Isaac, that's actually very kind from you, and that's always I mean the case when you have so kind introductions is at some point they become embarrassing. So thanks a lot for being so generous. Probably I don't deserve that, but thanks a lot. It's very nice from you. What I would like to do is to present some kind of general summary of some of the things we've been doing in the last years, trying to understand actually how cognitive processing might work in a miniature brain, in this case, the brain of a honeybee. A way to introduce this problem would be directly related to what Isaac said about the kind of general thinking we might have when we consider different kinds of brains. For instance, this is a frontal view of a human brain with an impressive volume. You can see the values there, and an even more impressive amount of neurons. And if I show you at the same time a frontal view of an insect brain, specifically a honeybee brain, and of course the drawings are not at the same scale, just by looking at the numbers, you will realize the huge differences between these brains. 1 cubic millimeter and 1 million neurons, actually less, it's 950,000 to be more precise. And then indeed, people might be tempted to think that endowed with such a little brain, insects might be rather machines or exhibit low levels of plasticity. So one question that I face typically is, why do you care about learning, memory, cognition, and why would you care about those using insects? Of course, we know very well that insects learn, and people in your university, like Martha Weiss, by the way, have been very influential to show learning in butterflies, and many other people have shown learning in other insect models. The reason why we like bees actually is that the honeybee forager that you see in this picture, actually all these foragers have developed learning and memory capabilities in a natural context, I mean in the context of forager. So these guys show the so-called flower constancy, meaning by that foragers, honeybee foragers, remain working on the same species for days eventually, if and only if these species provide profitable nectar reward or pollen reward they are looking for. So it means that they remain constantly working on the same species if it remains profitable. And the reason for this flower constancy is simply the fact that bees learn and memorize the features that allow them to find these exploited food sources. They learn the color, they learn the shape, they learn the odor, and they learn the position in space. And in space, that's what allows them to return constantly to the same place. The other reason why bees are attractive is that they are experimentally accessible, meaning by that you can train them. And you can train them, for instance, to fly into the lab and to search for a drop of sugar water, which is the equivalent of the nectar they are looking for. And the bee will be doing hundreds of flies between the hive and the lab just to collect that sugar water. And in that way, she will be able to answer questions that you will be asking. Like, for instance, how is your memory for this color? Do you remember 3 days later? Can you make the difference between these two odors and things like that? The answer of the bee will be encoded in its behavior. And they are extremely cooperative, meaning by that as long as the sugar water drop is there, they will be flying all day long without any break, just to answer your questions, meaning to work in your setups. The other advantage is to speak about this little brain. We say that it's a small brain, it's a relatively simple brain. I put simple in quotes, as you might realize here, because I mean simple in terms of the amount of neurons, not necessarily in terms of its capacities, as you will see. And the interesting thing is that contrary to other insects, many invasive approaches have been developed through decades to access this brain. We have imaging, we have electrophysiology, we have different possibilities to access this brain while it is learning and memorizing. And you will see some of them today. The other reason why we like this piece, and that makes a difference with other insect models, is that besides simple forms of learning, simple associative learning that you might find in almost all insect species existing, or nematodes or whatever. I mean by that simple association between one stimulus and its consequence, or a behavior and its consequence. Besides that, you will find other things in these little creatures. You will find higher order forms of learning, and I will speak better about this briefly. I mean by that, forms of learning that cannot be explained by these very simple associations. You require a further level of complexity to understand what's going on. And these, for instance, even if we use, just to give you an example, even if we use Drosophila in my team for answering some questions, we don't find them necessarily in Drosophila. Okay, so this is a particular appealing aspect of the bee behavior, and I will give you some examples to know what we are talking about. So what are the questions I would like to ask today? So I was telling you that we know that in natural conditions, bees learn and memorize different kinds of information, colors, odors, shape, and so on. But the question is exactly the one I raised before. Do they show just simple forms of learning? Say the simple associations one-to-one between, say, a color and reward, another color and punishment, odor, reward, and so on. These simple links, is that just the whole story? Or can they exhibit some plastic behavior that people would not hesitate in calling it cognitive or higher order processing if they would see it in another animal, like a primate, for instance? And if yes, actually, for instance, Examples of these can be the so-called nonlinear discriminations, we will speak about them later, or conceptual learning. And if they have such forms of learning, how do they happen in this little brain that you can see here? What are the circuits, the mechanisms that allow them to take place? So can we find some architectures responsible for these forms of learning? These are the questions I would like to raise today. And probably the best way to start is to show you a couple of experiments, behavioral experiments with free-flying bees that will allow to see the complexity of the system we are facing here. So I would like to show you some experiments using free-flying bees that have been trained to fly into the lab, to solve problems within mazes, and which will reveal some unexpected capacities of this little brain. So basically what you do is you go to the hive entrance, you put some sugar water on it, for instance, on a Petri dish, you wait that bees discover it, then you displace the Petri dish to the lab, and the bees will realize they are in a new place, they will learn the features of that place, and they will start coming by themselves to this new place. And in this way, step by step, you can train them to go into mazes or boxes where you will present problems. So in doing that, the question I had long ago, because this is an old experiment, but it started the whole idea, was the question was whether they can learn relational concepts. What do I mean by that? There are people like experimental psychologists like Thomas Sentel who define relational concept learning as a form of learning in which we learn relationships, he would say abstract relationships, encoded independently of the nature of the objects linked by the relation. What do we mean by that? I mean simply that we can learn to solve problems knowing that we have to choose, always say, the larger object. It's not important if the object is this or that. It's not important if it has this color or the other. It is used for this or that. The shape might be different. It's just that you should actually get rid of all this information and understand that the rule is choose the larger one. You can also be trained to the other way around, which is choose the smaller one. Okay, you can also be trained to another kind of relational rule, like choose the one which is above, or choose the one which is below, or choose the one which is at your right, at your left, and so on. We are able to solve these problems. And typically what we do, first we concentrate on the objects until we realize that the objects themselves, I mean, their physical nature is not important, but the relation that binds them is what counts. So this is why it's called a conceptual learning. So this is exactly what I wanted to know at that time. I wanted to know whether bees would be able of this level of abstraction, so getting rid of the objects and learning a rule. And I focus on a rule which is called the rule of sameness, or the principle of sameness. And in this particular case, you train an animal, the subject, in such a way that the animal has to learn the following rule. Choose what is shown to you irrespectively of what has been shown to you. Basically, you show an object. I don't know if you're seeing me right now in your screen, but for instance, look, this is my glass. I show you this glass. You just look at that. There is no reward, nothing. Look at that. Now, wait. And now I showed you these two objects. And now you choose. And now, of course, because the rule is what has been shown to you, you have to choose the glass. Next time, I might show you this, okay? And then I show you the glass again and this. But then you have to choose this, because this was what has been shown before. You choose what has been shown before to you. You see, the objects are not important. Objects are changing all the time. You have to abstract the rule of sameness. Choose the same. And this protocol is called the delay matching to sample problem, a well-known studied problem in vertebrate literature, cognitive literature. And the way we did it with the bee is we train a marked bee. So you can see, for instance, that the bee is marked with a green spot in the abdomen. So we know this is a bee. With our bee, we follow it all day long through several visits. And this is what we did. So basically, this bee was trained to fly into this Y maze. And we put a yellow disc at the entrance with a hole in the middle. So the bee, there's no reward, nothing here, just a hole. And the bee has to pass through the hole. And now the bee sees two possibilities, a blue disc and a yellow disc. Because the rule is the sameness rules, choose the same. It means that now it has to go to yellow land here, and only then she gets a drop of sugar water. If she goes to blue, it's wrong. And of course, it gets expelled from the maze. He has to do the whole thing again. It's a punishment. So 3 minutes later, the bee will be back from the hive, and surprise, actually, we changed the stimulus at the entrance. It's now the blue, which was the previously punished one. But this is not important because you have to use the sameness rule. In this case, the bee has to know that having blue at the entrance means that you have to enter and choose blue. So if you have yellow, you go and choose yellow. If you have blue, you go and choose blue, the sameness rule. And of course, this is done in such a way that, I mean, you get rid of left-right effects and so on, okay? This is nevertheless not enough to show an abstract rule. What you have to do is to confront the animals now with a fully new situation, telling you if they have a rule or not. So after training them consistently with this yellow-blue problem, at some point, we show them this, which is achromatic patterns, vertical or horizontal. So there are two possibilities, broadly speaking. So either the animals arrive here and see this for the first time and they are totally disoriented, or they have a rule. And if they have a rule, they wouldn't care. They would say, okay, vertical at the entrance, then I have to go in and choose vertical. And if it's horizontal at the entrance, I have to go in and choose horizontal. We had another group of animals which was trained on the patterns and tested on the colors. And of course, the other group trained on the colors and tested on the patterns. This is what happened in this experiment. Here you see the training. So these are blocks of 10 visits to the maze, meaning by that 60 times the bee flying back between the hive and the lab and making choices in the maze. And you have two curves, bees trained on colors, blue, blue, yellow, yellow, and so on, and bees trained on patterns, vertical, vertical, horizontal, horizontal, and so on. You see that at the end of the training, just to give you an idea, that takes probably 1 day of training. The bees are quite good, actually. 70% of correct choices, 75%. It's not bad for a bee, actually. But now comes the critical moment. At this moment, we show them the novel situation. So bees trained with colors here arrived and saw a vertical pattern at the entrance for the first time, or a horizontal entrance, the sample. And so when they saw vertical at the entrance, these colored trained bees went into the maze and in the very first choice, choose the vertical pattern. And if it was horizontal at the entrance, they entered the maze and choose the horizontal option. Bees trained on the patterns, okay, now arrive and saw for the first time the blue disc, and they entered the maze and choose blue. And if they saw yellow, they went for yellow. So this experiment was interesting and it shows you the complexity of the system, as I said. We are here far away from simple associative learning in which the animal just rely on what he knows. Here we have an animal transferring a rule between known stimuli and unknown stimuli from a different modality. I don't have time to show you here, but in that paper, we can even show the transfer between odors and colors and use of the sameness rule in these two different modalities. We also show in that work that they can also learn the opposite rule, which is choose the contrary to what I'm showing you here. I show you yellow at the entrance, and you have to understand you have to go to blue, and so on. And they have no problems in learning that rule too. So you see why it's called abstract learning, and you see why we are here far away from blue means reward, and that's all, okay? The simple associative learning. Since then, of course, we have done many, many experiments to understand and characterize this relational conceptual learning. And we have shown many, many, many things, like, for instance, this nice story by one of my students, Aurora Vargas Weber, where bees were trained in such a way that they had to combine two rules together, that the two objects had to be on top of each other and not just left or right of each other, and also that they had to be different from each other. And you might use different colors for the first time, and if they were placed in the right position, the bees would choose them, transferring the rule again between modalities. But I underline here this one, because this is the transition to the next story I want to tell you. One of the rules we were able to show they could learn was the rule less than. So we knew that we could train them in such a way that if we presented, I don't know, a certain number of items here and another number of items here, they could be trained to go always to the lower quantity, okay? We knew that. And of course, this was the basis for some experiments showing that they have a numeric sense, that they can count. Meanwhile, again, no time to tell you about these papers, but we know that they can count until 4. Some people say 5, but eventually definitely 4 or 5. It's not much, but that's definitely a counting ability that has been shown in the case of bees. The story I want to tell you, which is very recent, is done by a young postdoc, Scarlett Howard from Australia. She already left the team and she has now a position in Melbourne. And she did that in using this learning or this choosing or understanding that you have to choose less than. And she wanted to know if bees would have something like a 0 concept in their numerosity, in their sense of number. What is 0? Okay, there are different levels we can use to define 0. 0 might be just nothing, the absence of everything, okay? You can elaborate a little bit more by saying, okay, yeah, but what is nothing? Nothing exists because there might be something in an opposite way. So it's nothing versus something. You can elaborate a lot, actually, or even more, if you would say that, well, 0 is something more than this broader concept. 0 is a quantity at the low end of a positive series of numbers, 0, 1, 2, 3, and so on. This is already an elaborated definition for 0, okay? And at least it incorporates the sense of number, the fact that you know that 0 is the basal quantity in a positive series of increasing numbers. And of course, there is another definition that you would not expect in bees because it requires the symbolic representation as an Arabic number. This one, for instance, okay, that's the way we use to describe 0. Of course, the bees do not write and would not have a way to elaborate a symbolic representation, of course, but that's what's used in modern mathematics and calculations. But what do bees have? Do they have something like this? Do they understand 0? So what Scarlett did is this very funny experiment in which she trained, again, free-flying bees to fly into a maze. In this case, there's nothing at the entrance. The bee has just to enter and make a choice. If you look well this image, you see that here there are 2 squares and here there are 3 squares. She knew that this would work. I mean, she knew that it's possible to train bees to go to less. So in this case, bees have to go to 2. In this case, they get reward. If they go to 3, they don't get any reward. They are expelled from the maze and so on. The trick here is that the training is done in such a way that the numbers would change all the time. But the principle of having one option always being less than the other was kept constant. So numbers change all the time, stimuli change all the time, different, not only squares, circles, triangles, and so on. But I mean, the principle is the same. You have to choose always the lower quantity if you want to get the reward. So for instance, just to show you some examples of the stimuli she used, here you see the 3s, for instance, diamonds, squares, circles, the 4s, the 5s, the 2s, and so on. She controlled for low-level cues, density, area, and so on with many controls. But the training is what I explained to you. This is just an example. Here, for instance, 3 versus 2. The red square here means that there was sugar reward here, but there was no red square, of course. It means this is the one you have to choose. Then 2 versus 4, then go to 2. 1 versus 2, then go to 1, and so on. Training was very long, like 50 trials, if I remember well, but at some point came the test, and the tests are the exciting part of the story. Because here she showed something she never, ever used during the training. She presented 1 versus a white wall, nothing. With the question, what would do bees in this case? They know that the rule is choose less than. Would they go to the white wall because it's less than 1, something like the equivalent of 0, or would they be disoriented in this case? And she repeated the test by presenting 2 versus 0, I mean 2 versus the white wall, 3 versus 0, 4 versus 0, and so on. Like here, 4 versus the white wall, and so on. to see what bees would do in this fully new situation. Here's the answer. You see the proportion of choices for the white wall, for the equivalent of 0, okay? And here I'm showing you this test, 0 versus 1. You see that, and this is significant, they significantly prefer to go for the white wall that they never saw before and to avoid the 1 number in this case. What happened in the other test, 0 versus 2, 0 versus 3, and so on. This is what happened. Not only was that the same trend observed and it was significant, but you can see that it become better and better. And this is what people in human numerosity call the numerical distance effect. What is that? It's simply the fact that we have a hard time in distinguishing numbers that are very close to each other, 2 and 3, for instance. But it's very easy for us to distinguish numbers which are well separated from each other, 2 and 20. Well, it's a piece of cake, very easy to distinguish these two. 2 and 3, more difficult. 0 and 1, you do it. And 0 and 6, more easy or easier, as you can see here. Okay, numerical distance effect. So this experiment is also interesting because, again, it breaks some preconceptions. I mean, these are not robots. These are not simple associative machines. These are animals with unique capacities in terms of conceptual learning, numerosity. And you can see this experiment shows that for them, this white wall was the equivalent of something lower than 1. And you can see by the increase in performance, when you get, you are more distant from that 0 value, they become better. There's no time again here, but Scarlett could show in some of her last papers that they can even do some primitive summation or subtraction, addition, and very basic, of course, but the capacity would be there at least. Sometime this should change the perspective we have about these animals. Yes.
Can we interrupt for questions?
Anytime.
Heidi, do you want to ask the question or do you want me to read it?
Yeah, sure. No, I'll go ahead and ask. This is fascinating. Thank you so much. And my question was, how well can bees learn if the rule is imperfect? Like, can they pick up on strong patterns? If you throw in an exception to a rule, does it derail them and they go back to ground zero in training? Or can they accommodate a rare exception?
Yeah, that's an excellent question. We never did that. What we did, but that's not your question, it's reversal of the rule, which means having trained a rule, we retrain the animals to go for the opposite rule and to see how plastic they are, how long does it take. And we show a learning to learn effect. So being good in the first rule accelerates the learning of the second rule. We compare that with learning of the second rule from the very beginning, we see the slope of learning is totally different. But what you are mentioning is a different experiment, is to somehow, if I understood well, to intersperse within the training of a rule an exception. That we never do. We never did, sorry. That would be great, actually. We need to do that. Yeah. That's the only thing I can tell.
I'm always happy to give other scientists more work to do. So, that's great.
You did. Thanks. We might speak about that later, perhaps, because I would like to ask you a couple of things about that, if you don't mind. Great. So if I may continue, I don't know if there were other questions.
Oh, you can go ahead.
Okay. So I was saying that this changed the perspective about these little animals, at least, or should at least, or that was my goal, at least. And it tells you why, when I was telling you, okay, we use Drosophila in the team, but Drosophila do not offer this level of complexity, at least so far. And this is why make bees attractive, okay? So, and this is when people ask you, but why do you care about bees? You're interested in higher order cognition. Well, some of the answers have been already provided by these examples, I hope at least. So, but of course, for a neurobiologist, a behavioral neurobiologist, that's not enough. The experiments I told you are fascinating, at least to me, because they show the full power, the cognitive power of the system, but they do not tell you how this is taking place in the brain. As a neurobiologist, you would like to know. And these are free-flying bees. You cannot open the head of a bee and of a flying bee and run behind to record what's going on, not now at least. And so you need other preparations to access the neural basis of cognition. So, and to access these neural mechanisms, you need protocols in which the animals actually learn and memorize, and you can elaborate some different forms of learning in those protocols. But the basic thing is that the animals should not move. Because if you want to record neural activity, of course, the animal should stay quiet, because otherwise your electrodes or whatever move and you get noise and you don't get the right signals. So one of the things we use in the lab is this very famous protocol that many labs use also, which is a case of Pavlovian conditioning, in which you have harnessed bees, here. You see that the bees is immobilized in small metal tubes. Only the head is going out from these tubes, and which provides this facility of having a bee which does not move practically. And what you're doing here is a case of olfactory conditioning. The previous cases were cases of visual learning. Here we are studying olfactory learning. And what I have here is a proboscis. So the proboscis is the tongue of the bee. And this tongue is normally hidden, you don't see it. But there is a reflex, an innate reflex. When you touch the antennae of a hungry bee with a drop of sugar water, boom, the proboscis goes out. This is the proboscis extension reflex, okay? It's an innate response to sugar on the antennae. Tack, the bee extends the proboscis because it means sugar should be around. I better extend my tongue to lick it. And here what we are doing is we are pairing the presentation of a neutral odor, an odor that does not elicit these response, with the sucrose stimulation, so that the animal creates a new association between odor and sugar. Basically, you have your animal immobilized in this little tube. Here you have an olfactometer sending a constant flow of clean air to the animal, in which you can introduce a pulse of odorant with control properties, timing, and so on. And right after the odorant, you will provide, for instance, with this primitive way, just a toothpick soaked in sucrose solution. You will provide the sucrose to the antennae, the bee will extend the tongue, and you will allow her to lick the sucrose solution. In doing that, you will have created this association, odor means sugar. That's the way it works. Have a look here. So, sorry. Here you have the bee seen from above. This is the harness, it cannot move. Only the antennae and the mouth parts can be moved. And you will see here a red square showing the first time an odorant is presented to this bee. This odorant has no meaning. You will see no particular response to the odorant. Right after that, we will give the sugar. So the animal is there, and the odorant will arrive, as you will see now. No response, sugar in the toothpick, and you see the proboscis extension response. The animal licks the sugar, was very stimulated. You see the proboscis is still out, looking for more. And by doing this, actually, as I say, you have created this association between the odorant, the conditioned stimulus, as Pavlov used to say, and the sucrose reward, the unconditioned stimulus. And the proof that it works is that now you will show to the same bee just the odorant in the absence of sugar. And when you do that, because the odorant means sugar, because the animal has learned the association, now the animal extends the proboscis to the odorant in the absence of sugar, because odorant means sugar. This is a simpler form of learning, very simple associative learning. And of course, what we can do is to make it more complex, to ask questions about cognitive processing in the bee. We have to understand this protocol first in its most basic form to understand what's going on here. This is a very, very robust and fast learning. You have seen just one trial, I mean by that one odor-sugar pairing, and you might have here your response of a group of bees showing proboscis extension response, PR, to an odor. Let's call it odor A+. Plus means that it got reward. And if you have 1, 2, 3, 4, 5, 6 trials, you typically get a response curve like this. So after 2 trials, typically on the 3rd trial, most bees have already learned the association. So 80–90% of the bees have learned that. If you consider that trials are separated just by 5 or 10 minutes, you will see that it's incredibly fast. Not only that, if you keep these bees in an incubator in the darkness, avoiding any kind of further odorant during that time, just feeding them with sugar but without odorants, and 2 weeks later, okay, you bring them to the setup and say, okay, guy, now you're back here. Do you remember which odorant you got here 2 weeks ago? Was it A, B, or C? Typically, the responses you get are similar to ones. So this is why, as Isaac said when he introduced this talk, bees were a traditional historical model for understanding memory properties. And the advantages that of this system is that, I mean, because the cuticula is quitting, so it's a non-innervated material. You can expose the brain by cutting a hole, and you can record, for instance, neural activity in the brain of this learning bee while it learns or while it forms memories. And that's a big advantage because the bee will be behaving normally despite this situation. The only thing you have to care in this case is that you have to perfuse regularly your brain to avoid desiccation by being exposed to the air. By doing that, I mean, you might record during learning, during memory retrieval, and so on. There are several works, I mean, done in this direction. And for instance, in the last one we did last year, we showed that they are incredibly fast because a single pairing of odor and sugar, we show triggers already molecular cascade leading to protein synthesis from, and then therefore to the establishing of long-term memory. So we believe that several trials were necessary for that. Now we show that one trial is enough. So they are incredibly good at forming memories. So we can have a look at this.
Can we interrupt again?
Yeah,
we have a question from Afia. Afia, do you want to go ahead and ask, or do you want me to read the question?
You can go ahead.
Okay. Okay, I'll read it. She's asking if has training bees given an insight into their attention spans and how that can provide any knowledge into its mechanisms in humans. So attention.
So how was that to train the bees with what? Sorry.
Yeah. So she's asking if this kind of training or behavioral paradigms in bees has given any insights into their attention spans.
Yeah.
And how?
That's a very good question. There are several experiments. There's a whole line of research on attentional processes in bees. By the way, also in Drosophila, as Isaac might know, Bruno van Swinderen has been doing a lot of works in attentional processes. You use paradigm which are different from these. For instance, a typical paradigm that people use for studying attention is to have, for instance, two competing percepts. Okay, you have, for instance, one moving bar here and another moving at a different frequency here. And so in the animal then has focusing attention on one or on the other, and then you have this competition between perceptions. And people have, I mean, shown that by using these and other paradigms that we can speak about attentional processes in insects, and not only that, but which are the neural substrates. And we know that some specific subsets of dopaminergic neurons are engaged in these attentional processes. And there's, as I say, a lot on attention in insects. Think about the protocol of delay matching to sample. Of course, the protocol is not done for that, for tackling the issue of attention. But at some point, the animal has to get rid of accessory information to focus on what is the rule providing the solution to the problem. So it needs some attentional focus at some point. But of course, I'm not using this to speak about attention because there are specific protocols to test attention. And it suffices to say here that they have been used also in bees and in flies, and bees and flies respond in the way primates or humans do in these attentional paradigms. I hope I am answering the question in this case. Okay, so coming back to this olfactory learning, I just show you, let me show you what do we have here, because this will be the last thing I would like to tell you. and I was discussing that with Martha West before, and which is the fact that we humans have different brain regions that might be dedicated for different forms of learning. For instance, we know that the hippocampus and the prefrontal cortex that you can have here is the hippocampus actually, are necessary for specific forms of complex learning. For instance, hippocampus might be involved in different forms of spatial learning, but not necessarily on cue-based learning, so learning that a cue is followed by a consequence. It might be necessary for problems like categorization concept learning in the case of the prefrontal cortex, and other forms of simple associative learning might not require these structures, okay? They might be solved elsewhere in the brain. That's a principle common to the human brain, that we have dedicated structures for certain levels of problem complexity and others doing other kinds of problem solving. And so the question we had here is that we would find some organization principle, cognitive principle, some similar principle in the case of the bee brain. So, and to do that, I'll show you how this olfactory brain is organized. So we are speaking about odor learning. So what happens in odor perception is that you have odor molecules arriving at the level of the antennae. The antennae are the real nose of insects. Okay, so the antennae will stimulate olfactory receptors on the antennae. You have approximately 60,000 olfactory receptors in the case of bees. Then when these receptors are stimulated, they will send a message through an antennal nerve to the brain telling I'm smelling this or that. The first structure in which odor processing takes place is the so-called antennal lobe. You see one here and the other here, antennal lobes. These antennal lobes are built in such a way that they look very similar to the olfactory bulb of vertebrates because they are done of subunits called glomeruli. And from there you have projection neurons, meaning by that neurons that leave this structure where the first olfactory encoding took place to bring this process message to higher order areas in the brain, in particular to the so-called lateral horn, diffuse, badly ill-defined structure here, and then, of course, to the mushroom bodies, which are these huge structures that occupy 30% of the brain, which have been historically associated with long-term memory, storage and retrieval. So you see an olfactory brain with different levels of processing. You start the receptor level, you move here, you move here, you move here, you have parallel tracks. So it's a structured brain in terms of olfaction. So if I would just show you one hemisphere, this part of the brain, okay, this is what you see. So this is the antennal lobe, this structure, you come from the antennae here. You have tracts going to the lateral horn here. You have tracts going also to the mushroom bodies, but other tracts going this way to the mushroom bodies, these big structures you have here. So it's a complex olfactory circuit you have here. Not as complex as in a mammal, of course, but nevertheless, it's not a simple story what you have here. And here comes my last experiment. I don't want to stay at this level of odor sugar learning because this is too simple. I wanted to know if in this paradigm I can establish complex associations too. And I asked the question whether bees can learn so-called nonlinear discriminations. And if yes, which brain structures would be required for these? So what are nonlinear discriminations? Nonlinear discrimination is a kind of higher order form of learning in which you have something like these. Let me illustrate with this example. These are euros, of course. I use euros because I cannot do it using dollars. You will see why. Let me ask you the following. So Isaac, I see you now on the screen. So imagine I train you in such a way that anytime I show you stimulus A, you get from me 100 euros. Okay, not bad, right? It's like $130 or something like that. And anytime I show you stimulus B, you also get 100 euros. So there is A, 100 euros, then it's B, 100 euros, and so on. What would you expect if I show you now AB?
$200, 200 euros.
Exactly. Okay, that's a perfect answer. And that's the way we reason, actually, because what we do is automatic summation. It means that we process these problems in linear terms, okay? A means 100, B means 100, then AB is A plus B, linear processing. It has to be 200. You see, we have a note for 200. So this is why we use euros. So it works well. That is what would you expect. However, this non-linear discrimination, what you do is to train a subject in such a way that he needs to understand that this automatic summation, this linear summation, does not apply. So it gets 100 euros on A, he gets 100 euros on B, but anytime I show you AB, there will be nothing. So the subject has to inhibit this linear processing until he or she understands that AB means nothing. So we conceived this protocol using the proboscis extension reflex with one of my postdocs, Nina Deisig, and a colleague in Germany, Harald Lachnitt, who is a famous experimental psychologist. And we did in such a way that we trained the animals with this so-called negative patterning problem. The negative patterning problem is what you have seen already. It means you get stimulus A, reward, stimulus B, reward, AB, no reward. It's called negative patterning because the pattern AB is negative. It's simply that's the reason of the name. And it's a difficult problem, really difficult, because if your tendency is just to focus on the elements, on A and B per se, you're lost, because each element is as often rewarded, A plus, as non-rewarded, A minus, B plus, B minus. So if you just focus on the elements, you don't get the solution. You have to understand that A in the context of B, or B in the context of A is something different from A alone or B alone. So it requires a higher level of complexity. So how it would work this in theoretical terms, it would mean that in blank trials, your animal does not respond. When you have odor A rewarded, then the animals extend the proboscis. You have an odor of these machines pointed toward the antennae, you provide odor B, and then there is proboscis extension, and then they should learn that when the two machines are sending these two odors simultaneously, separately, but simultaneously, I mean, the animal should inhibit the response. So it should learn that AB is different from A plus B. We knew that this is a difficult problem because some of you might think, oh, but I mean, perhaps in the brain, AB is X, something totally different, and then the problem is trivial, okay? Well, we knew that this was not the case because we did some imaging of the antennal lobe. So to understand how odors are processed and coded, we did some calcium imaging, meaning by that you put some calcium dyes here, which bind to calcium release within active neurons. And we can see the active neurons responding to this or the other odorant and see how an odorant is encoded in this structure. By doing that, we knew, for instance, that processing of binary mixtures is linear. So have a look at this example. This is the response you get here to one hexanol, meaning that this area, which is called glomerulus, this region of the antennal lobe is responding to hexanol. If we provide linalool, we can see activation here and some activation here, you can see. Now, if we provide both simultaneously, we get this pattern. Now, forget this pattern and let's do the following. Let's take these two and let's make a theoretical sum pixel by pixel of these two images. When we do this, we get this, which is, I mean, exactly this. Of course, it's not by eye that you have to do some correlational analysis, you do some principal component analysis, and you show that they are highly correlated. So it means that the brain of the bee tends to process A, B as the linear sum of A and B. So then it's difficult for them to do this because we are asking to do the opposite, okay? So we knew that this would not be a trivial problem. So what did the bees in this case? So we wanted to know if these structures, the mushroom bodies, were necessary for solving this kind of problem. And what we did, you see, these areas are fascinating because not only because people say that this is the place where long-term memories are stored, but because there are multimodal regions. Here you have arrival of olfactory information. Here you have arrival of visual information from the visual regions of the brain. Here you have mechanosensory information, gustatory information. This is, as we say in French, a carrefour a place of convergence of multiple afferences. And all this information is combined within the structures, because when you record the neurons at the output here, these neurons are multimodal, responding to odors, colors, light, and so on. So this is a place of combination. So if someone would ask me, and this would be just a wild speculation, how is it possible that UV transfer a rule from color to odors? Well, this could be the place. I don't know how, but at least I know that color and odors can be combined and integrated here. Okay, not all mushroom bodies of all insects are like this, because this is a fully multimodal mushroom body. Some mushroom bodies of other insects are dominated by one modality, just olfactory, or mostly olfactory, or mostly visual. This one is unique in the sense that it has a kind of equilibrium between different modalities and integration of these modalities. Okay, so we wanted to know actually if this structure would be necessary for this higher order non-linear olfactory learning. And to answer this question, we blocked the mushroom bodies. So we injected into the mushroom bodies, and you can see here a local anesthetic. We injected procaine. So procaine is what your dentists use to avoid pain. So, and we knew that we did paper before to characterize the mechanism involved in this procaine use. We know that it blocks voltage, gated sodium and potassium channels. So what happens is you block the mushroom bodies during a particular time, which we define. We knew how long the effect will last and so on. And then you get bees that are active, behaving actually, but do not have functional mushroom bodies. So it's like have a patient with a lesion in the brain and you ask him, what can you do? Can you do this? Can you do that? In this case, the question is, can you do a linear discrimination? So this is just the way you do it. Sorry, you can see here that the windon has been cut before. You can see here the cut and has been replaced. The animal is behaving normally, as you can see, despite the operation. Then you take it away to inject the procaine. Now you can inject the procaine here and here. You cannot see probably, but with our trained eye, we know where are the calyces of the mushroom bodies, where is this place. And we will use some staining to, with appropriate molecular weight to show that it stays there. You can see the little spot of the drug here, you can see probably, and on the other side. So these mushroom bodies are not functional anymore. Then you replace the cover. You see the animal still behaving normally. As you can see here, you replace the cover, and the animal is ready to go, okay? And you can ask him now, what can you do? And here's the behavior. So you can see these are control animals that have been injected with saline solution. No effect on mushroom body function. And this is already a nice result because you can see that these animals learn to respond to A+ and B+ separated. Here I pulled both responses. And they learn to inhibit the response to AB-. See, they have a hard time at the very beginning, but then they learn. And they learn to respond to A+, B+, but not to the joint presentation of both. So they are A of this nonlinear problem solving. And now I will show you just the end of the training to avoid complicated learning curves. It's the same. So here you see they are learning more to A+, B+, and not to or less to AB-. Okay, what happens if you inject procaine and you kill, in quotes, the mushroom bodies? No discrimination, no way to learn the problem. From this, you could conclude that mushroom bodies are required for this higher order problem. But I would say, stop. No, you need a control. Perhaps procaine is just killing the olfactory sense, generally speaking. You don't know. You need to show me that if you show a simpler problem, they can do it even if the absence of mushroom bodies, if your argument is that mushroom bodies are required for a complex problem. And so that's exactly what we did. This is the work done by my assistant, Jean-Marc Deveau, in the team. And he said, okay, let's train them with A+, B+, CD-. You see, it has a similar structure to the previous problem, but this is a piece of cake. But each stimulus has a single non-ambiguous association. This one is always rewarded. This one is always rewarded. This one is always non-rewarded. This one is always non-rewarded. Simple associations solve the problem. A good, B good, C bad, D bad. Okay, here, I mean, it's simple. So indeed, bees injected with saline learn the discrimination very well. They respond to AB+, and practically not to see the minus, even better than before in the other problem, as you can see. And what happens with the procaine injected bees with no mushroom bodies? They also did it. Okay, so it means that in the absence of mushroom bodies, you can still learn to solve simpler problems, elemental problems, these simple associative A good, B good, and so on. But you cannot learn a non-linear problem which requires inhibiting this treatment AB equals A plus B. Okay, so in the absence of mushroom bodies, the question would be, well, where these does happen? So we have some preliminary evidence, but not conclusive, that they might use the antennal lobe, the upstream structure, to solve this problem. It doesn't mean that for a simple discrimination, they don't use the mushroom body. If it's there, they will use it too, of course, and they will store memories and so on. But it means that the mushroom bodies are essential for the higher order problem in any case. So then we did this thing, which is, we knew that there were these two populations of feedback neurons in the mushroom bodies, the A3D neurons and the A3B neurons. This is the equivalent for Drosophila people of the APL neurons, because these are GABAergic feedback neurons. And so here are the schemes. So this neuron takes information at the output of the mushroom bodies, goes back to the entrance and delivers GABA, so inhibits the entrance. This one does the same, but does it from the output to the output, from the output to the input, from the output to the output. So we injected here or here picrotoxin, which is a GABA inhibitor, to see if these or these neurons were necessary for this kind of problem solving. So this is what you can see here. So this is where we injected, here at the entrance or here at the output. Okay. And what we did that before the training, of course, and this is the result. When you inject at the level of the lobes, so these neurons from output to output, okay, you can see that the bees still learn the problem. They respond more to A plus, B plus, and less to A, B minus. So killing this tract of neurons or the GABAergic signal at this level didn't change anything. What happens if you inject now here at the input region, affecting this other population of neurons? In this case, the animals were not able to make the discrimination. So it means that from these two inhibitory mechanisms, these ones are the necessary to solve the problem. Why inhibition? Because somehow you have to inhibit that you respond to A at some level, to B some level, and then you would add both. No, you have to inhibit that. And perhaps this is the inhibitory mechanism that you require, provided by these A3B neurons to solve the problem. So you can see that the interesting thing is that we have two lessons here, that we have a brain that shows a similar principle, the dedicated principle that we find in higher brains, higher order brains, mammalian brains, structures that might be used to some kind of problem solving, other structures to other problem solving, and so on. Okay, that's not unique to more complex brains. We see that mushroom bodies are required for these nonlinear discriminations. And we even see, we can even trace some mechanisms responsible for that. And that's the newest story. This is what we are doing now. Look, what is the combination between the first part of the task visual learning and the olfactory learning? Well, I told you it was not possible to record from the brain of flying bees. So we created this virtual reality system. And that is what we're doing now. So look at here, this is a bee that is set on, it's tethered from the thorax. It cannot escape, but it can walk stationary on this ball, which is floating on an air cushion. And his movement will make the panorama move. When she goes to the left, the panorama goes to the right, and so on. So she actually creates displacement and so on. And depending on its choices, she gets reward or she gets punishment from us. See, for instance, this is 3D. She sees these two blocks here. One will be rewarded, the other not. And basically, she would see something like this at the end. This is what we reconstruct from what she does. She's going towards one, then she changes, and then she gets reward, and then she will actually learn. that she has to go to the green one, okay? And already we did the same experiment, the negative patterning, using this, for instance, A plus, B plus, AB minus, and we show already that they can learn it also in this modality. They respond more to A and to B and not to AB minus, as you can see here. And now we have some talented people who are, you can see here the same system, the virtual, the ball, the bee walking and so on, the 3D landscape. And this is under multiphoton microscope. So we are imaging brain activity while the animal is learning this, or we are using multi-electrodes to record what's going in the brain, what's going on during this problem solving. So I'm done. Sorry if I've been a little bit long. So these are my general conclusions. So I try to show you that this brain is not stupid, of course. It's not simple. Remember, I say simple in quotes in terms of the number of neurons, but not in terms of the complexity of the behavior it produces. Of course, it can produce stereotype behavior, reflex responses, and so on. We have seen PR, for instance, but there is plastic behavior, and this plastic behavior goes beyond this simple associative learning. We have seen forms of conceptual learning, we have seen numerosity, we have seen nonlinear discrimination. So this brain is not primitive, it's not a rudimentary brain. They are not just associative machines, there's more than that, okay? And interesting thing is that it's not just that they are more than that, but we can go deep into the mechanism and understand some architectures and principles to grasp what's going on when these creatures solve these problems. So yeah, so you see, I mean, of course we have Drosophila and we love Drosophila, but when you use non-traditional model systems in neuroscience, you can learn other things. Not best, not better, simply others, okay? And that is a real stimulation to me, at least. Let me finish just by showing you this guy. I think it's important. I mean, sometimes we should acknowledge the shoulders on which we stand, and sometimes we forget to do it for political reasons. I mean, one of the things we discovered when during the lockdowns of COVID, we had for the first time a lot of time to read and explore things we never explored. And we knew, of course, that there was a guy somewhere lost in the literature that, who was called Charles Henry Turner. We didn't know anything about his life, like you read papers and do not know anything about the authors themselves. The funny thing about this person is that actually he did some experiments who were forgotten by anyone, by all the people in the science community. But the interesting thing when you go back to these experiments that we're speaking about 1890 or the beginning of the 19th century, he was already speaking about learning and memory in insects at a time in which nobody was speaking about that. The story is particularly sad because Charles Henry Turner, I didn't know that at the time, was the first African-American scientist who get a PhD from the University of Chicago. Not only that, he published almost 70 papers. I discovered that much later. Actually, I knew only a couple of them, and I tend to dismiss them as old stuff, actually. When I returned and read these papers, I was totally baffled because, I mean, this guy was speaking about memory, learning. All the people were ignoring that. People ignore him later. Why? Well, the reason why he was ignored is that he had a tragic destiny, actually. He got, as I say, 70 papers or more, 3 of them in science, by the way, okay? And he was never able to get a position in any academic institution in the USA. Why? Simply because he was Black, okay? And there are documents proving that he was dismissed and forgotten just because of his racial origin. And he ended up as a teacher in a little school for Afro-American kids in Missouri, if I remember well. And even then, he tried to do experiments and publish papers. And most of them were forgotten until he was rediscovered recently. So we wrote a paper about him. And I think it's necessary in these times to recover this part of scientific history we are sometimes tending to forget. And in this case, I like to mention him because you can see the link. I am speaking about cognition, learning, memory, but there have been valuable people before me, even if we tended to, or we didn't know that. And it's good to give them the place they deserve. Okay, so that's all. This is the people who deserve the credits. I mentioned, for instance, Jean-Marc Deveau, who did the imaging experiments. He's somewhere here. This is Scarlett, the zero woman, and so on, and other people. And thanks to you for your attention, and to Isaac for the invitation.
Thank you. Thank you. We have some questions. Megan, do you want to ask the question, or do you want me to go with it?
I can ask. I actually have two questions, if that's okay. So the first one is what I put in the chat. So you were talking about, I think in the second to last part of your talk, about like how it just takes like those 6 training sessions in order to induce like lifelong learning. And I was wondering if there was any manipulation to be done after those training sessions that can prevent them being able to remember it afterwards. I'm just thinking, I know like in humans, you have like hippocampal memories that are transmitted to the cortex, so they essentially become like independent of the hippocampus. And I was wondering if you see that as well.
Definitely. So for instance, what you can do in these experiments is inject, if you are asking for a memory that they are building, you can train the animals. So you know that they've been able to solve the problem because you see how the learning curve separates. But right after the training, you inject, for instance, as we were discussing with Marta West before, protein synthesis blocker. And in this case, what you are doing is to somehow inhibit the process of protein synthesis, which is required for forming long-term memories. So the animals would have learned, but they would not keep a memory of that. That's a basic procedure you can do. I think this is what you are asking, if I understand you well. Other things could be done. One of the things, for instance, that could be done is to, right after the training or during the training, you could use distractors. Okay, so for instance, to combine the training, odor, sugar, would say some distractors like flashes of light or something like that. The question would be attentional then. Would a distractor, for instance, somehow interfere with the learning of the olfactory association? You might argue, yeah, well, I mean, the olfactory tract is well separated from the visual tract, so it might have no effect. But then I should remind you that somehow these tracts are combined in the mushroom bodies. So perhaps for some problems, the integration will be destructive and then it will interfere with your learning. Depends on the problem you're addressing. So yes, these experiments are possible, and some of them have been done already. And it's a lot of fun actually to do them.
Okay, Afia has a question too. Yeah. No, go ahead, Megan. I forgot you had 2 questions.
Sorry, that's okay. My next question was, I know that like bees are really social creatures, and so I was wondering if you isolate bees before you do any sorts of these like learning tasks, if they show like any inability to learn.
Fascinating question, and you touch on a point that was for many, many years my big frustration. Because the big frustration here was to say, okay, learning and memory have to be studied. That's a norm, okay? At the individual level, you have to use individuals because you encode your experience in your brain, and this is your brain as individuals. So you need to study how you learn individually and how you memorize individually. And the frustration was, okay, but these are social creatures, okay? How I'm getting rid of the social component totally because I'm just using isolated individuals, okay? Because this is how a learning experiment has to be done. But I mean, what do I losing by doing that? And so we found a way to solve at least partially this problem. And you might see we did quite some papers in the last 2-3 years on that. We incorporated the social dimension by asking which effect would have on this learning and memory individual performance, which effect would pheromones have, social cues. So what we did in all this line of research, we did several kinds of exposure to pheromones of different meaning and different valence. By valence, I mean attractive or deterrent ones and so on. And the question was, would the social signal change the way the animals learn and memorize? The answer is yes, it changes totally. We have some papers and we showed that exposure to pheromones, if they are appetitive pheromones, pheromones that are used in the context of say foraging, for instance, and you then after that, and long time after that, so it's not just immediately, you train an animal with odor sugar association, which is also appetitive learning, then performance gets better. It's somehow that the exposure to the appetitive pheromone prepares the animal to learn better about an appetitive problem, okay? If you expose the animal to an aversive pheromone and then you give an appetitive problem, there is interference and you get worse. But if you expose an aversive pheromone and then you do aversive learning, odor shock, then they also get better because there is concordance between the signal and the learning. So that's the way we answer your question.
Great. Afia, do you want to ask your question?
I think he kind of touched on it already.
Okay.
Basically, I was just asking, does different odor preferences or exposures in bees influence how they learn or process information?
Yeah, well, Afia, that's not bad also because part of your question is not the same. You are asking about odor preferences, which is not necessarily pheromone based. And because there are indeed, as in many insect species, some odorants that bees prefer. I mean plant odorants. So these are not pheromones, okay? And we showed that, for instance, these preferred odorants, for instance, not being sugar reward, they are able to elicit the proboscis extension reflex. So you give the bees these 2 or 3 odorants. We know one of them is linalool, the other is phenylacetaldehyde or something like that. I don't remember the exact name. But I mean, they trigger already the proboscis extension response being odorants, okay, without any sugar. And we can show that also bees might be prepared to learn better about these odorants, you see? And here we're not speaking about pheromones. So there seems also to be some preparedness for some signals so that they might learn better some sensory signals than others. The same applies to colors. When I was a postdoc, one of the things I did long ago is to show that there are some specific wavelengths that they learn better than others. And together with a good friend who was also doing a postdoc at that time with me, Lars Chitka, and he was more ecologically oriented than me. He showed, look, let's sample thousands of flowers in Europe and with spectrophotometer, and let's measure the reward. And it turned out that the flowers that were more associated with good reward were exhibiting these wavelengths that the bees preferred in the very first flight. That is an old work we did together long ago. But you see, your question touches on these preferences, and it's very good.
Great, thank you so much.
Thank you.
We have one last question from Grace. Grace, do you want to go ahead and ask?
Sure, thank you so much. It's been a really interesting talk. And in a lot of your graphs, it looks like the sort of learned response peaked somewhere between like 70–80% or so. And you mentioned that like if bees were learning a new rule, that they learned the second rule faster than they had learned the first rule. And I'm just wondering if you've looked at any, if there are bees or individuals that completely fail to learn, and if you
that's
looked
an
into
excellent
that.
question. Yeah, that's an excellent question, and you're totally right. Only recently we have started to pay attention to this point, because actually we had a kind of automatic way of doing this experiment. We say, okay, let's take only the good learners, and because they will provide the perfect answer to what we are doing. And okay, there is 20% of bees that did not learn. Okay, I don't care about this. They are in the graphs. True, I'm not just picking my bees, obviously. They are in the graphs, but I'm not asking questions about these 20 or 30%, okay? And you're right. And only recently we started focusing on this variability in learning performance. If you have a look, actually, you will see that this week, precisely this week or last week, there's a paper we published in Journal of Experimental Biology, which addresses this problem. The problem is, or the question that this paper asked is, okay, we have good and bad learners. Is a good learner proficient in all kinds of problems and sensory modalities, yes or no. If I have a good learner in a visual discrimination, and now I take it and bring it to olfactory discrimination, is it also a good learner? And if I have a bad learner in the first case, is it also a bad learner in the second case? Is proficiency maintained, yes or no? And not only that, is proficiency maintained between levels of complexity? So simple problem, conceptual problem is one, and that we showed. And we showed, basically in this paper, we showed that proficiency is maintained between levels of complexity. So if I have a bee that is good at solving a simple discrimination, it will be essentially good to solve a conceptual problem. But proficiency is not maintained if you change the sensory modality. A visual learner is not necessarily a good olfactory learner and vice versa. So it's very exciting new world for us that we are trying to study now.
Very interesting. I think we are. Wait, do you have a question, Heidi? Or is what the same, right? It's what's related.
No, it was just it was related to Grace's. I was having a similar. I was validating that I thought her question was awesome because I had the same one.
And if I might ask you, if you could, I mean, this is to Heidi actually. The idea you mentioned of doing this kind of exception to the rule, if you would have some protocol published or something that you could provide to me to think a little bit more about adapting something like that, it'd be very exciting. So Isaac has my mail. And if you would come with some idea, I mean, I think I would love to do that.
I feel, Martin, I'm about to let you down in a big way here. The last time I really thought systematically about bee behavior and learning was when I was an undergrad taking courses. I'm a parasitologist and I do completely different research. So it was sadly just a random idea that I'm
okay.
going to have to leave completely in your hands. But it was great. This is a fabulous talk.
Okay,
thank you.
credits have to be. If we do something, you get the credits at least.
Well, thank you, everybody. We are way out of time, but thank you, everybody, for coming. Thank you very much, Martin, for coming and talk about your research.
Thanks to you. Thanks for your patience.
Thank you.
Thanks a lot. Bye-bye.
Bye.
Thank you.